We've decided to make the capstone project optional. If you do not want to participate Friday because of the holiday, that is perfectly fine. It will not be counted against you for the coveted certificate purposes. We'll still count it as complete participation. So what we're going to do is by the end of today, I would like you to send me your group and the data set you'll be working on. And if you do not want to participate on Friday because of the holiday, just send me an email and let me know. No questions asked whatsoever. So we will still continue as we planned and we set out at the beginning of the week. We'll just have an option for you guys to sit out to observe Juneteenth. Should be straightforward, but are there any questions about that? No? Okay. And Yanan is going to send out an email about that and post on the Slack regarding that option. Yeah, good. Thank you, Chad. Okay, so I am going to share my screen and we'll introduce the topic for today and go over the lab from yesterday. So I again forgot to put a .pdf at the end of the file name. Sorry about that. So the topic for today is going to be multiple testing and clustering. But I'm not going to start there. I'd like to briefly go over the lab from yesterday. So we went through the DSeq analysis with the Pasilla data set here, and the model we're running is type plus conditions. So we're adjusting for this the type of analysis that was done. It was either paired end or single read. And then we're adjusting for treated versus untreated. That's what our p-values will express the difference there. There are some highly significant genes that pop up here with some incredibly low p-values, adjusted or otherwise. At 0.05, just over 1,000, 0.01, 735. The change from those significant at the 0.05 level. 592 were downregulated, 488 were upregulated. So a little bit more downregulated than upregulated. And it seems this, I don't know, maybe that makes sense. I don't pretend to know the science of that. And we see some information here about the most significant genes, the top 10 genes, the size factors, how they varied. So this is based on the library sizes from 0.65 to 1.8 about. So some difference there. The largest is over 2 times, almost 3 times as big as the smallest, but those are things that we can adjust for. The dispersion plot, which we talked about in good detail yesterday, the red line being the fitted value, the black lines being what is estimated just on the gene-specific data, and the blue line being the final value, which combines those two. Here, this was the code that I gave you for the dispersion estimates for 3 genes, the gene with the highest mean, the gene with the lowest non-zero mean, and the gene with the most significance to it. And we can see the dispersion value is a lot lower for the one with the highest mean and the one with the lowest mean, which is, and this is typical, the dispersion values will be higher, and the top differentially expressed is in between those two, certainly closer to the lowest one. We have the volcano plot and kind of the ones we want to pay attention to. Top left corner, top right corner. Those are ones that have high significance and large log2 fold change in absolute value. Those that have looked at how many are significant and are either upregulated or downregulated. How many are significant in the middle? These ones in the center are ones that we would not want to pay attention to. Those are ones that have small log2 fold change. Upper left, 113, upper right, 109. So in terms of how many are up and down regulated that are actually important, ends up being relatively similar. And then we have a heat map here, clear relationship between those that are upregulated, downregulated, and how they are expressed, overexpressed, underexpressed, how that affects, how that is relative to treatment. So this one, which I didn't ask you guys to do, it compares the model with versus without library type. So this one only has condition in it. And when we first did the principal components, we could see that type seemed to have a pretty big impact. The adjusted one that adjusts for type, it does find more significant results than when we don't adjust for condition. So there is more power. It is a good thing to do to adjust for it. This is a comparison of the top 20 genes in terms of their log2 fold change. Really no difference at all here. So the genes that have the 20 most significant p-values, their log2 fold changes are almost identical. So the adjusting for condition gives us a little more power. We get more significance when we do that, but it doesn't change the log2 fold changes that we get from the analysis. So overall, it doesn't have a large impact in that respect. And this is a comparison of the MA plots. They look very, very similar to one another. So, and this is just a little discussion of that. So adjusting for type, it is a good idea. It does give us more power, but it doesn't overall have a huge impact on what we see towards the end of the analysis. Right. Any questions about the lab? Okay. All right. So with that, we'll go towards the what we have today. Where is it? Okay. So today we'll talk about multiple testing and clustering. These are two things that multiple testing is something that happens directly in the DSeq function that we're using. Clustering is something that we were looking at in those heat maps. So we'll just talk about both of these issues in a little bit more detail, what they are and how they work. So when we run DSeq2, we get two columns that matter a lot in terms of the significance. We get the raw p-values and the adjusted p-values. They both come out. Today, we'll talk about how these are different, which one to use. Of course, the short answer is always use the adjusted one, and talk about are there any other options of things that you can use. So first, let's just introduce the multiple testing problem. So in a standard hypothesis test, you would threshold at alpha equals 0.05. And when you do that, there's a 5% chance of a false positive when H0 is true. So if we're just doing one hypothesis test, that's fine. But what about when we're doing 20,000, for example? So to look at this, let's go to the HTML document for today. And this is going to start with a demonstration of this idea with the airway data set that we've been using. So here I'm loading in the airway data set and creating a DSeq object with the counts, the sample info, the design matrix, and then setting the referent level for treatment so that the log2 fold changes will be in the way that we want them to be. We are going to filter, so we only keep those that have at least 10 reads per over all the samples, run the DSeq, and then get the results. So if we look at the genes, first off, how many were tested? So how many non-NA p-values did we have? We have over 22,000. If everything was null, so if there was no signal at all in this data set, how many would we expect to have a p-value less than 0.05? Well, that's going to be 0.05 times the number that we tested, which will be 1,118. So if there was no signal in this study, like the treatment had no effect on any of these genes whatsoever, and we cut off the p-values at 0.05, we would expect to say that over 1,000 were significant. Those would all be mistakes in that hypothetical situation. And that's the problem that we have with the raw p-values. Now, why would we get this many mistakes? Well, when the null is true, the p-values have what is called a uniform distribution. They have a uniform distribution. So if you look at a histogram of the p-values, when the null is true for all of them, it's going to be just a flat histogram, okay? There's not going to be any spikes in it or anything like that. The probability that a null p-value is between 0 and 0.1 is the same as the probability it's between 0.1 and 0.2, the same as the probability is between 0.2 and 0.3, so on and so forth. If we looked at a histogram, as I said, we would think it would look uniform. Now here, this is a histogram of the raw p-values. It does not look uniform at all. We can see that there are many more p-values in this lower region. So many more p-values that have a small value that are in this the smallest or second smallest bin. Now, once we get up here, we can see that the heights of these bins are relatively similar as we go across. So that's that null uniform distribution that we see. The adjusted p-values do not have a uniform distribution. So it's not that, it's the raw p-values that have a uniform distribution when the null is true. And the fact that we see this spike down here, that's an indication that there are a lot of signals in this particular study, which we have seen from the results that we've already looked at. So if we cut off at 0.05, we're going to get a lot of false positives. What's the probability that we get one false positive? It's going to be equal to 1 minus alpha, all to the power of m, where m is the number of hypotheses that we do. So if we do 20,000 tests, the probability is 1 that we make a mistake. This figure shows that probability. So down here, if we do one test, it's the probability we make one mistake is 0.05. If we go up to 20 tests, the probability that we make at least one mistake is over 60%. If we do 40 tests, the probability we make at least one mistake is about 85%. And then once we're up to 60 or 80, the probability is basically 1. So if we do enough tests, the probability we're going to make a mistake is almost certain. So as statisticians, we don't like this. We don't like that, that we don't know what the probability is, that we're not setting that. So there are some methods to control the error rate in these situations where we have multiple tests. So to control the error rate, we first have to talk about what error rate we want to control. There are two separate error rates. We'll talk about the family-wise error rate and then the false discovery rate. Okay, the family-wise error rate. This is the probability of at least one false positive. When we control the family-wise error rate, we control the chances of getting at least one false positive. So commonly when we do something like this, we may control the family-wise error rate at okay, it's not going to be. Let's go here. Okay, I get it. Yeah. All right. Sorry, some technical difficulties here. Yeah, I get it. There's two figures. Okay, let's see if I can do this. Okay, so if we control the family-wise error rate at 0.95, say, what we are doing is we're saying the probability that there is, excuse me, not 0. Do this backwards. Let's say we control this at 0.05, not 0.95. I'm doing one minus. So let's say we control the family-wise error rate at 0.05. What are we saying? We're saying that we are very confident that all the results, among all the results we got, they are all correct. So if we reject 100 hypotheses with a family-wise error rate of 0.05, we're very confident that all 100 of those are correct. There is not a mistake among the 100 that we do. So that is a very high bar to reach. This is going to lead to family-wise error rate procedures being conservative. They don't reject a lot, because the more you reject, it's a higher bar that you have to have. If you reject 1,000, we have to be very, very sure that there is not one mistake among those 1,000. That's hard to say. So these types of procedures, they end up being conservative. Where these are used mostly is in a clinical trial, say, involving 3 primary endpoints. So in that situation, all right, we have a clinical trial we're running. We have 3 different outcomes we're looking at. We want to control the family-wise error rate. We want to make sure that if we say all 3 have an effect, we're very sure about all of those statements, the collective of all those statements. Okay, we don't want one false positive in there. That's where we're going to use family-wise error rate in situations like that. The other error rates, the other common error rate, these aren't the only two, but these are the two most common, is the false discovery rate. The false discovery rate, commonly we're going to control this similar to before, 0.05. And what this says, if we control this at 0.05, it says that among all the rejections that we make, about 5% of them are mistakes. So if we reject 100 hypotheses, we are, the false discovery rate is going to say we're confident that only 5 of those are mistakes among those 100. So we got 95 that are probably the real deal. 5 of those are probably not. Compare that to family-wise error rate. When we reject 100, we are very, very sure that all 100 of those are the real deal. So it's a much higher bar for this family-wise error rate compared to this false discovery rate. And so this is something that's more appropriate when we're testing many, many, many hypotheses, and some false positives are acceptable. So if we're doing an RNA-seq study, identifying candidate genes for follow-up, a few false leads are tolerable. This is not going to, the FDR is not going to be as conservative as the family-wise error rate. So if we apply both of these procedures at the 0.05 level, the family-wise error rate may reject 6 hypothesis where the false discovery rate will reject 30, right? So that we're going to have a lot more rejections for this false discovery rate because it's not as high a bar as the family-wise error rate is. When we're doing a clinical trial, if we have 3 primary endpoints, we want to be sure that all 3 of those statements we're making in the end are correct, that they are true. We don't want to say a treatment is effective when it's not. If we're doing an RNA-seq study and these are things that we're following up, a few false positives are okay. So that's why the false discovery rate is common in these types of studies. Okay, any questions about family-wise error rate versus false discovery rate? Okay, we'll move forward. So if we want to control the family-wise error rate, the most common method of doing this is called the Bonferroni procedure. It's very simple. Here, what we do is we divide the alpha by the number of tests. So if we do 100 tests, so if our alpha is equal to 0.05, the Bonferroni level we would compare the p-values to would be 0.05 divided by 100, so then we get 0.005. So very simple procedure to implement, and it is a very low bar. I mean, it's a high bar. The p-values have to be very small to reach that number. We can get adjusted p-values. So the p-adjust that comes out of DSeq is similar to what we see here, where we multiply the p-values by the number of tests. We cap it at 1, of course, because p-values can't be greater than 1. And then these adjusted p-values, we just compare to the regular alpha level. So the unadjusted p-values, we would compare to this adjusted alpha level. The adjusted p-values, we would just compare to the regular alpha level. That's the difference between adjusted and unadjusted. So for example, if we have 20,000 genes, alpha equals 0.05, a gene must have a raw p-value below 2.5 times 10 to the negative 6 to be called significant. When do we use this? Usually we use this when we have a small number of planned comparisons where any false positive has some pretty serious consequences. Don't really use this in genomics because the threshold is so stringent that almost nothing passes and many true signals are missed. So using this for genomics, I wouldn't say that it's being cautious. It's making a different kind of error because you are missing so many signals that you end up having a situation where the results are they're not great. They're not what you'd want. So we'll look at the threshold for the data that we have, and what this ends up looking at, how many reject, and what we get for this Bonferroni procedure. Okay. Okay, so how do we control the FDR? We use something called the Benjamini-Hochberg procedure. This is one of those highly cited papers that first developed this procedure. It's a pretty simple method. There are many methods to control the false discovery rate. This was kind of the first one that introduced the false discovery rates and showed how you can control it. So this is how it works. And of course, you don't have to do any of this. DSeq calculates the adjusted p-values for you. There's also another function in R called p.adjust. It will also calculate these for you. So this is not anything you have to do yourself, just showing it so you have some conceptual understanding of how this works. So the different thing about the Benjamini-Hochberg versus the Bonferroni is that you don't compare every p-value to the same threshold. When we do Bonferroni, everything gets compared to alpha over m. When we do Benjamini-Hochberg, they aren't all compared to the same value. What they are compared to is a function of their rank. So first thing we do when we do the Benjamini-Hochberg procedure is we sort the p-values from smallest to largest. Well, and then we find the largest k such that the kth p-value is less than or equal to k over m times alpha. So the smallest p-value we would compare to the Bonferroni alpha, 1 over m times alpha, or alpha over m. So the minimum p-value is compared to the same threshold that we use in the Bonferroni. The second p-value is compared to that Bonferroni threshold times 2. The third p-value is compared to the Bonferroni threshold times 3. The kth p-value is compared to the Bonferroni threshold times k. After we find that largest k, we reject all hypotheses that their rank is less than k. Similar to before, we can get adjusted p-values from these. The formula is a little different of how we get the adjusted p-values. It's a function of k and the p-values that are greater than it, but we can get adjusted p-values from this Benjamini-Hochberg procedure. So for you as a practitioner, you will get the adjusted p-values. This is, and you'll compare those to 0.05. This is a little look behind how those adjusted p-values are created. It's the same formula that we had before, just divides by k for the kth p-value. Basically that. There's a little more to it. But this is going to be far less conservative than the Bonferroni procedure, clearly. Only the minimum value is compared to the same threshold when you do that, when you employ this one. So here is an example of this. So we have 10 p-values here. The rank, excuse me, we've sorted these from the smallest to the largest. Here I have included the threshold, just the k over m times alpha. So the first one, this is the same as the Bonferroni threshold. 2 times the Bonferroni threshold, 3, and so on and so forth. So for the first one, is it less than this Benjamini-Hochberg value? Yes, and it would be rejected. This one, is it less? Yes, it would be rejected. The third one is not less. So this is where it gets a little bit interesting. The third one is not less, but we actually end up rejecting the third one. Why do we reject the third if it's not less than that particular value? That's because the fourth one is. The fourth one is less than its value. So we reject it. And the way the Benjamini-Hochberg procedure works is you choose the largest value that is under the threshold and you reject everything above it. So a particular value can be above the threshold and still get rejected. And then from rank 5 all the way on down, we don't reject. So here's a little look graphically at how this could work. So here is a p-values. These are all sorted. We have 10,000 p-values. And this is all of them. On the right, we're only looking at the first 15 or so. The line here is the Benjamini-Hochberg threshold. So we can see that the first 11 are all below, and then we are above after that. So in this case, we would reject those first 11. Now, if this 10th one was above, say right here, and the 11th one was, it has to be higher than the 10th, but maybe just a smidge higher, and it was below, say right here, we would still reject the 10th and 11th because that 11th is less than the threshold, even though the 10th isn't. Okay, let's take a look at these two procedures in our airway data set. So the Benjamini-Hochberg, this is going to control the family-wise error rate, the probability of at least one false positive. The FDR is going to control the false discovery rate, the rate of mistakes. So here in this situation, our alpha at 0.05, number tested. Here's the threshold that a hypothesis would have to meet to have a rejection. So when we use the Benjamini-Hochberg, when we use the Bonferroni procedure, we would still reject 1,413. So we still reject a good amount with the Bonferroni procedure. If we compared the unadjusted p-values to 0.05, we would reject 5,600. So the Hochberg procedure, the BH procedure will be between these two. It'll reject more than the Bonferroni, but it'll certainly less than the unadjusted p-value. And this is this example that we just went through, the sorted p-values compared to their threshold. We take all those, we would take the first 4 because we find the maximum value that's less than that threshold and reject everything above that. This is a graphical look at what those p-values look like. And again, we reject up to 4, even though 3's p-value is above that threshold. Let's take a look at this in for the data, the airway data. So another way to get the Benjamini-Hochberg p-values is to use this p.adjust function. It comes out of DSeq2, but we could calculate it this way. How it works is we give this function the unadjusted p-values. We say, all right, adjust these p-values according to the BH procedure, and these will be the manually adjusted p-values. Here, creating a table that has the adjusted p-values from DSeq and the manually adjusted. and we see that there's actually no difference here. This is not, excuse me, the values here are identical. It's not always going to be the case. There's some little differences that can happen from, rounding errors and things like that. But in this situation, we actually get identical p-values from the manually adjusted in DSeq 2. How many do we reject? Looking at the three criteria here for the raw p-values, we would reject 5,600 tons, way too many. The BH rejects 4,000, so 1,600 less than the raw p-values at 0.05. The Bonferroni all the way down at 1,400. So among these 4,000, there are about 5% that are mistakes. It's about 200 mistakes that would be in here. 200 false positives among the 4,000 that we reject. So some singly, certainly sprinkled in here. Among the Bonferroni, those top 1,400, we are very confident, 95% sure that there's not one mistake among those top 1,438. So Bonferroni rejects far, far fewer than the BH does. I'd like to work this back into our what we were looking at yesterday. I want to look at let's see if I can make this. Actually, I'm going to open this up. I would like to look at this figure again and see how this, I'd like to see how this volcano plot looks different with the Bonferroni adjusted p-values versus the BH. So I'm going to go all the way down to this, click this volcano plot. I'm going to hit this down arrow. That's going to run everything above it. And this is just so we can look at this in a little more detail. So, I mean, this study has so much significance to it, and so it's a little hard to we find so many significant genes either way, and I wanted to redo this table. I'm not going to look at the figure. I wanted to redo this table where we're looking at the log fold. So that's 4.6, interpreting top hits. I'll go here, interpreting top hits. There we go. And this time, all I'm going to do is I'm going to add the regular p-value to this. This may do nothing. I haven't looked at this. So these results, they don't have the regular p-values to them. So I want to add the unadjusted p-values to these results. Rerun this. And then I'm going to look at, let's look at the top 20 in terms of log fold change. And I'm going to add the regular p-values here. So that will allow us to see if any of these that are in the top 20 of log 2 fold change and are significant with the BH, would they be not significant with the Benjamini-Hochberg procedure? And it's only giving me a little bit of this. So I'm going to use the view function so we can look at this look at this in more detail, and we see that is the case. So the threshold that we need to meet 0.05 by about 22,000. I can't remember the exact number, but we'll just, so yeah, it was 2.5 times 10 to the negative 6. So this gene right here, you can see its p-value is 1.1 times 10 to the negative fifth. That would not be rejected under the BH procedure. And there's even some higher, even this one right here, would not be rejected under the Bonferroni procedure. So this is a situation where we have a gene that had a very large effect size. Its p-value was not as, its unadjusted p-value, it's not significant at the Bonferroni. Oh, that one is. It just barely. I guess we got to go down to this one. Same thing though. So high log 2 fold change, its p-value would not be significant under the Bonferroni. So this is one that we would not find. We can probably see it. I think it's this one down here. So has the fifth highest log 2 fold change, but is not significant at the Bonferroni. Is there probably a true signal in this gene? Yes, there probably is. I mean, the Bonferroni is a very, very high bar. So we have to be 95% sure that there's not one mistake among all the tests that we make. It's a very, very high bar. So this one probably is, it probably is a true signal. There's probably some difference between the treated and untreated. Has a very large, robust log 2 fold change, and okay, we would miss it. So that's kind of the difference between the Bonferroni procedure and the BH procedure. We would miss some of these situations where we have a large log 2 fold change, but for one reason or another, we did not end up getting a p-value that got to that very, very low level that we need, that 2.5 times 10 to the negative 6. And I'm sure there's some down here. Yeah, most of these down here, it has to have at least e to the negative 6 to be significant. So all these down here are not going to be rejected under the Bonferroni. All of these are rejected under the BH. So just wanted to give you a little kind of look at what the actual difference is in terms of the hypothesis you're really studying, you're really looking at. You could miss out on some important signals when you're using the Bonferroni procedure. Okay. Any questions with that? Okay, go back to the notes here. So, family-wise error rate versus false discovery rate. What are they controlling? One is the probability that there's at least one false positive. The other is the expected percentage of false positives or proportion. Setting that these are used, Bonferroni, when there's a few plant tests, they're all very important. False discovery rate, many simultaneous tests where we're making any mistake is okay, that we can have some false positives in there. How conservative are they? Family-wise error rate is very conservative. BH procedure is still moderately conservative procedure, but it certainly has a lot higher power than the Bonferroni procedure does. Family-wise error rate, it is used in genomics sometimes, but not all that often. The BH procedure is used much more frequently. Clinical trial, you'll often see the Bonferroni procedure there. But sometimes when they have a lot of different things they're looking at, the BH procedure will be used. It's gotten to be a lot more accepted in practice. Um, and I was going to show you this, something I was looking up yesterday. Um, Sometimes I've mentioned that some of these things that we've gone over are some of the most highly cited papers in science. I was interested. What are the most highly cited papers in science? So I looked this up, um, and number 11 on here is the Kaplan-Meier curve that we talked about last Friday. These are from the Web of Science. And you can see over 38,000 times cited. So we've gone over that one. The Cox model, which we also went over last week on Friday, one of the most highly cited papers. A couple others, statistical papers pop in here. And then at number 59 is the Benjamini-Hochberg procedure that we talked about today, which has about 16,000. This is a year old or so. And these rankings are, of course, changing all the time. But to show you that this has been a highly influential procedure and it is very, very widely used. Um, okay. So DSeq2, it applies the BH procedure automatically when you call results. There are other procedures that you could use. So I'm going to go back to sorry, I got some other stuff open here. I'm going to go back to the what we've been using for today. I'm going to get that .qmd file open. And you may have to install some of these packages as usual. I'm going to go down to where we did the manually created. There we go. So this is where we manually adjusted the p-values using the BH method. The BH method is not the only approach that we can use. So there are some others. So if we look at question mark p.adjust, you can see some of the other methods that are here. The Holm, Hochberg, Hamel, and Bonferroni. These are all these are all methods that control the family-wise error rates. And then we have the BH and Benjamini-Hochberg. That's another one that we could use. The FDR is actually going to give us the BH adjusted p-values back. So there are some other methods that you can apply here to adjust your p-values, but always give this function the unadjusted p-values when you want to do that. Some other things that DSeq does. We talked about this a little bit. The independent filtering. This is going to get rid of genes that have very low mean expression levels, going to exclude them. Testing them would require extreme evidence and would only inflate the multiple testing burden. So when we're using the Benjamini-Hochberg or we're using the BH procedure, the number of tests we do plays a big role. So when we do that filtering, we decrease the number of tests that we do, and it makes the multiple testing, a little bit easier. It doesn't have a huge impact, but it's going to make it a little easier. And what we're getting rid of are tests that we likely wouldn't find anything anyway. So this is going to increase the power. So that's the thought behind the independent filtering. DSeq2 also does some outlier detection. So genes that have extreme counts and get flagged under this outlier, they end up getting p-values equal to NA. So that they're also thrown out. Both these procedures are conservative. They'll only increase the power for the genes that are remaining. They never create false positives. The downside to them is just that we have a lot of genes that we didn't test, we didn't look at. So those need to be put aside for another day. Okay, so that's what we have for multiple testing. That's a good place to stop for us. So why don't we take a break and we come back here at 11:05 and we can continue on and talk about clustering. All right, see you back here in about 10-15 minutes. Okay, we'll continue on here with just talked about multiple testing. We'll continue on with clustering. I'm glad I had a little chance to take a break there because I gave me some time to figure something out that was confusing me in our code in the example. So, which I'll share that with you. So we talked about multiple testing, something that's going to be useful to tell us which genes are differentially expressed. But in this situation, we get tons of significant results. Even if we just got 500, it's hard to interpret one at a time. So what we end up with, let's say we're interested in the top 500. We have some way of identifying the top 500 in some fashion. What we have is we have the counts for 500 genes among treated and untreated. What are we going to do with that? Clustering, this is where clustering comes in as a method that we can use. Clustering is what's called an unsupervised learning technique. It takes data and puts that data into groups. There's no outcome when we have clustering. So here we have treated and untreated. The clustering is not going to use that. It's just going to use the expression profiles among the subjects. So if we give it 500 expression profiles, it's going to use that to create groups, clusters that we could then take a look at. How we're going to use it here. Can we group genes with similar expression patterns across the samples? So rows of the count matrix, which rows are similar to one another. We can group those and then we can say, all right, what's going on with this group? What's going on with that group? What's going on with that group? We can group samples. So we can group the genes. We can also group the samples with similar overall expression profiles. Here, we would obviously think these samples would group by treatment. So grouping the genes is more of interest, but we can do it on samples as well. In other studies, like the Framingham Heart study, for example, there we may be more interested in grouping by the participants. For the genes, for this genetic data we're looking at, probably grouping by genes would be more scientifically meaningful. So we'll talk about two clustering methods. The first one we'll talk about is k-means clustering. So the goal here is to group all observations into k clusters so that observations within a cluster are as similar as possible. I'm going to show you how this algorithm works for this example data that I have right here. So this data, we only have two variables on it, and there are clearly three clusters of data points. So a nice simple setting. How clustering works is we pick. So let's say we're clustering these into three groups. How clustering works is all clustering needs to start somewhere. The k-means needs to start somewhere. And how k-means starts is it randomly selects 3 observations from the data. So let's start by saying, let's see if I can get some, let's start by saying we select a point here. Let me get this a little thicker so we can see it. We get a point here, maybe we get a point right here, and then maybe we get a point right here. So we randomly select 3 points. Then we group every data point in the data set with the cluster that corresponds to the closest of these green points. So for all of these data points, we would say which of the green points, these are called, the green points are called the centroids usually. Centroids. We have these 3 centroids. We say, all right, which centroid is closest to each of these points? Now, clearly, this would do a great job. All these points would be in this cluster, corresponding to that centroid. All these points would be in this cluster. Maybe this one would go down here, and then all these points would be in this cluster. That is step 1. Step 2, we recalculate the centroids. So we calculate them as the average among all those that are in the same group. So this centroid would then move from this spot over to somewhere around here, because that's going to be the average among all those in that group. This one would move over to here. This one would move over to here. Then we do the same thing. All the points again would get put towards the centroid that's closest, centroid that's closest, all the points down here. Recalculate the centroids, and we just keep going through that procedure until none of the points change hands. Now, an interesting thing here, let's say this point that we weren't sure which one it would go to. So let's say this point initially went towards the centroid over here in step 1. Well, in step 2, it would flip over to this centroid because this would come closer to this point, and then it would go over here. So really, after 2 steps, the procedure would be done, and everything would stop changing hands. So that was a good starting point because we got one centroid the centroids were in the 3 clusters. Let's say the centroids were, say, over here, I don't know, over here and over here. So this situation, it didn't work out as well. But for this centroid, all these points over here would be clustered to it. And for this cluster, it'd be about split in half. All these over here would go to this centroid. All these over here would go to that centroid. So this, when we recalculate things, everything would work out fine here. This one at step 2 would go there, and then it would stay there. At step 2, this would probably go, somewhere in between here. These would bring the centroid over, but these would restrict it from going completely over there. And then this line would sort of move this way, and we'd get fewer points that are going to this part. This one would move over. And at step 3, then this would move over even farther, probably, closer to here. And then at that point, this line would go, and then we'd be where we should be. So it's a really, it's a very simple procedure. And you see it written here. We randomly assign the centroids to start with, compute the means, reassign each observation to the cluster with the nearest centroid, and we just keep repeating these steps. What this algorithm aims to minimize is the within cluster sum of squares. So the distance between each cluster and its centroid, trying to make that as small as possible. Some of the things about k-means. We have to choose k first. So this is one of the central issues. So if we go back to this toy example here, what if we only chose 2? So if we only had 2, maybe we have a situation where we have points right here and here even. What would happen then? Would we end up with one centroid over here and one right here? Or maybe we'd end up one in between these two and down here? Unclear. So it would not work as well with 2. And also if we had 4, then we're having to split one of these clusters into 2. So when we do k-means, we do have to choose k. This is one of the issues. We'll talk about some methods to do that. We want to scale our data first. K-means uses Euclidean distance, which means that variables with large variance are going to dominate. So when you use the scale function, make everything have mean of 0 and variance of 1, for example, before we do any clustering with k-means. The results do depend on the initialization, which is done at random. So if we did that 2 example, so if we're doing, let's say we're doing this one with 2, if we start with one data point right here and one data point right here, we would probably end up clustering these 2 groups together, and this one would be by itself. If, on the other hand, we started with one data point right here and one data point right here, we may end up clustering these 2 together. We may end up with the same thing, but it is a little bit sensitive to where we start. So, and it's random. So the results are going to depend on initialization. It has a little randomness to it. K-means assumes that the clusters are spheres and the sizes are roughly equal. It doesn't work as well when we have clusters that are elongated. So if we had some data, so these are all pretty spherical, relatively good circles around these. There's not one of these variables that's stretching this more than the others. If we had a cluster that was, looks something like this, that's something we could see on the figure, but k-means wouldn't handle that situation as well. So that's another weakness of it. It is going to be sensitive to outliers. An extreme observation can pull a centroid toward it and kind of make things look different than if that outlier was there. Okay, so in terms of practical usage, always scale your data first and be careful of outliers because they can have a big impact. Okay, one of the main questions, how to choose K. There's 2 methods that we'll go over with. One is called the elbow method. The other one uses something called the silhouette width. For the elbow method, what we do is we plot the within sum of squares against K. So the within cluster sum of squares, that's our goal, is to minimize the within cluster sum of squares. So every time we add a cluster to our data, the within cluster sum of squares is going to go down. Okay, it's always going to go down. The elbow method, it looks, are there points? So here's a plot of the total within sum of squares in the number of clusters. The within cluster sum of squares is always decreasing. The elbow method says, look at a point in the figure where there seems to be diminishing returns. In this figure, it doesn't really appear to happen, but let's say the figure looks something more like this. You would say, all right, well, there appears to be an elbow there, so that kind of juts out a little bit. This is the number of clusters that I would choose. However, when you do this, a lot of times there's not a clear elbow. And so there's that, and it's a little bit subjective using the elbow method. The average silhouette width, this is a more deterministic approach. And what it looks at, we see BI minus AI over the max of AI and BI. This is the definition of silhouette width. On the numerator, we have the average distance of a point to the nearest other cluster. So in this example, it'd be like, all right, the average distance of this point to all the points over there, because that's the cluster that's nearest. That would be BI. AI is the average distance within between that point and the other points in its cluster. So what is the average distance of that point and all these points over here? This would be the BI, the distance between this point and all these. AI would be the average distance of this point and all these over here. We take the difference of those two and we divide by whatever one of these is largest, usually going to be the BI component. SI ranges from -1 to +1, higher is better. So if this is equal to 1, that means the AI is approximately equal to 0. That's closest to that. And we average this over all points, and it gives us some indication of how good the clustering is. We can do this for multiple values of K. You see this right here. Here's where we have 1 cluster, 2 clusters, 3 clusters, 4, 5. And this does not always increase. It can decrease. And here we see that 3 clusters seems to work pretty well. The 3, 4, and 5, they all look pretty similar, even 2 maybe. So there's not a lot of difference. There's certainly a big jump from 1 cluster to 2. But then not a lot of gain going further than that. So we kind of could use all these values. So it kind of also, this is kind of what we see here with the elbow method as well. It doesn't appear that there's like a great answer. If we had data like this, both of these figures would be very clear. The elbow method, you'd see a huge drop until we get to 3, and then it would level off there. And the silhouette width, we would see some more changes in that. So let's take a look at this with our data. So initially we're gonna set this up for clustering. Whenever we cluster, we usually want the data on a comparable scale. So here we're going to use something called the variance stabilizing transformation. And we're going to restrict our results to those genes that have the most variance. So we're only going to look at those genes that have the most variance. So we take our results, we apply this variance stabilizing transformation, and then we get the genes that have the top 200 most variance. So these aren't those that are most significant or anything like that. They're just the genes that have the most variance. We identify what those 200 genes are, and then we have here, this matrix will have the counts for those genes that have the most variance. Then we are still going to scale to have mean 0 and unit variance. So even though we've done this variance stabilizing transformation, we want to scale our data before applying the k-means procedure or the hierarchical procedure that we'll talk about next. Okay, so here is a look at how we will apply the k-means procedure. With k-means, we have to choose k. We have to give it the number of clusters that we are going to look at. So there's some very nice functions that we can use to do that. So this fviz_nb_clust is the one we are going to focus on. All of these analysis are going to use this scaled matrix of all going to be scaled variables with the top variability. So we give it that matrix. We tell it we want to do k-means. Here, how we are going to select k, we're going to look at the within cluster sum of squares. So this top figure is going to give us that elbow plot, the same one that we looked at in the slides. K max is going to be the maximum number of clusters that we're asking this function to consider. So it'll consider 1, 2, all the way up to 10 clusters. In start, this is going to be for each value of k, the number of random starts that it is going to use. So it's not going to do this once. It's going to do it 25 times for each value of k. We do that because k-means has that randomness to it. So we start using random values. So if we just do it once, we don't know, maybe the second time we would get very different answers. So here we do it 25 times, and this function is going to average over those 25 results. And then just adding a title. The second one, here's where we are looking at the silhouette width. Rest of the code is the same. So the only thing that's changed is here we have silhouette versus within sum of squares WSS. And these are the figures that we just looked at. So the elbow, we want to be looking for a bend in it where we really start to get diminishing returns. I don't see much of a bend. This seems very gradual to me. The average silhouette width there, we choose the K that maximizes it. 3 and 4 look pretty similar. Below, we're going to look at 4 separate clusters. Okay, so here we are going to fit k-means, and we are going to use the k-means function. Again, we give the matrix that's been variance stabilized and scaled and all that. 4 centers, so that means we want to make 4 groups. And again, here I'm doing 25 starts. So this is going to apply the k-means algorithm 25 separate times, and it's going to find the solution that had the lowest within sum of squares. So it's going to find which of those 25 appeared to work the best, and that's the results that it is going to give us. We get a table from this, which has the number of genes per cluster. So the first cluster has 35, the second has 91, the third has 45, and the fourth has 29. These numbers, 1, 2, 3, 4, they don't have any meaning whatsoever. They're just random. If we do this again, we could get the same results, and cluster 1 has 91, cluster 2 has 35, and cluster 3 has 29, and cluster 4 has 45. So the numbers don't really mean anything. And this is a visualization of the k-means clusters. So the data that we give it, it has 200 genes, 8 subjects. To visualize this, we're not visualizing it across all 200 genes. We are doing principal components, something we'll talk about tomorrow, to summarize those 200 genes into 2 dimensions that explain a lot of the variability. And we're seeing how these clusters look in terms of those values. So the biggest cluster is cluster 2. We see it right here. The yellow cluster here, cluster 4, cluster 1, and down here we have cluster 3. The circles that we have here, this is a little look at the distribution of those that are in this particular cluster. We can see the clusters that have a lot of overlap to them. We can see the clusters that are more unique. So in this situation, cluster 2 appears to be relatively unique for the k-means. It certainly has a high score on PC1, positive, and PC2 scores relatively close to 0. Not a lot of overlap. Clusters 1 and 3, much more overlap there. Clusters 1 and 4, good amount of overlap. Clusters 2 or 4 and 3 and 4 in clusters 1 and 2, they seem to have good separation between one another. Any questions on the k-means clustering?
Professor McLean, can I ask you a question?
Mm-hmm.
So like from the eyeball like picture, and then another one, it's say the k should be 3, and then how here is center is equal to 4, like k is 4 now. I mean, how do we decide to increase one?
It's just what I picked. Yeah, probably should have. Probably should have used 4. So probably should use 3. So let's redo it. I think I have this loaded in here. Okay, so that's how it looks with 3 clusters. Let's take one of these off. And so with 3 clusters, we end up with 52, 95, and 53. So the 95, that appears to be the same cluster 2 that we had previously. And then it seems like maybe cluster 4 maybe got sprinkled into clusters 1 and 3. We'll be able to tell more clearly on the figure. Yeah, so cluster 2, when we do 3, ends up being having a similar look to the way it looked last time. Make this figure larger. Yeah, we'll just zoom in. Cluster 3 looks like it took some of these from the previous cluster 1, and the rest of cluster 1 went with cluster 4. A few points up here that were in cluster 4 are now in cluster 2, so that's kind of why that one grew a little bit. But yeah, using the average silhouette width, that's what I should have used.
Okay, thank you.
Yeah, thank you. Good question.
Well,
Any other questions?
Can you hear me? I have another question as well.
Okay. Yeah, good question, Angel. Does the overlap indicate anything such that one needs to increase or decrease the number of clusters? It does a little bit, but not completely. And I'll tell you why. What we're looking at here are the first and second principal components. We are not looking at the full data. Principal components, there's the first one, there's the second one, there's the third one, there's the fourth one, there's the fifth one. And it goes all the way down. So here we could not see any overlap in it could be that there is a lot of overlap between 2 on the first, when we look at the first and second principal components, but then maybe it's on the third. Oh, okay, there we see why these clusters are different. So it's not definitive that if there's a lot of overlap in this PC1 and PC2 that there is a need for more clusters. But it's nice to see that, nice to see some separate clusters when we do this. Really, to choose the number of clusters, really these are our best methods, using the average silhouette or the elbow method. There's also something called the gap statistic, which is another thing to use. So when we see separation in this plot, it's nice. If we don't, it does not mean that we're using the wrong number of clusters. Yeah, I wish I could give you a more straight answer than that, but that's the way it goes. Great. Okay, let's talk about hierarchical clustering. This is sort of the second most common method, along with k-means here. So with k-means, we have to choose k up front, and it has some randomness to it. Hierarchical clustering does not have this. It does have some model choices, but you don't have to choose k up front. When we do hierarchical clustering, it builds a tree called a dendrogram, showing all possible groupings from n clusters down to 1. So it shows us the whole gamut of how we could cluster these variables. There's different ways to do this. The agglomerative, which is bottom-up algorithm, every variable starts in its own cluster. So we have, let's say, n clusters to start, and we find the two data points that are most similar. Okay, which two data points are the most similar to one another? We merge them into a cluster. Then we do the same thing. Do the same thing. Do the same thing until all the observations are in one cluster. So the result is a tree, like you see on the right here. This is the dendrogram. And you can cut it at any height to get how many clusters you want. Now, there is one question is, how do we tell how similar a cluster is from a data point? Or how do we tell how similar a cluster of 2 is from another cluster of 2? There are 3 different methods to do those calculations. The complete, which is the default, it looks at the maximum distance between any 2 points in the cluster. So if we have a cluster of 2 and 1 data point, we look at how far this data point is from both of these, and we take the maximum of that. This complete tends to produce compact, roughly equal size clusters. It's the default choice. Average, this says, all right, if we have 1 point here, 2 points here, we calculate the distance between those, we take the average. Single takes the minimum distance. So complete is going to produce roughly equal size clusters. Single is sort of the opposite of that. It produces long chain-like clusters. So we end up with one cluster with a ton of observations, and then we have a couple observations over to the side here. And average is kind of between the two. Most of the time I'll do complete. And then also with bottom up, where we start with each observation in its own cluster, there's also top down, where we start with everything in the same cluster, and then we start separating apart. Bottom up is more common though. So here's how this would, we have a dendrogram, example dendrogram over here. So when this is done in a bottom-up fashion, observations 19 and 20, those were the first 2 to be clustered together. So those observations were the most similar in the data set. And then observation 11 was grouped with them, creating a cluster of 3. That would be the second step. The third step, combine observations 72 and 30. And looks like we have 2 30s here. So 30 with parentheses, 30 without parentheses. I'm not sure how that works. But you can see how this goes along. So the height at which 2 points were grouped together is an indication of how similar those points were. If we want K clusters, we simply just would draw a line. So here, if we wanted 2 clusters, we would cluster all these data points together and these 3 data points over here. So we would get 2 groups with those. If we wanted more than 2 clusters, it gets a little tough from there, but 3, we end up getting pretty much the same thing. We get all these in one cluster. 59 is in a cluster by itself, and then these 2 are clustered together. If we want 4, it's hard to see what happens first. Do these 2 split apart, or does that split apart? But anyways, you get the point of how we can create clusters from this. Yeah, so let's take a look at this with our data, and this is where I have a mistake in here that was confusing me. We'll talk about that. I figured this out during our break. So we're going to cluster. First thing we're going to do is cluster the samples, and this is where I made a mistake. So to do this, we do this in 2 steps. We're using the scaled matrix again. That is not what we're going to give the h-class function. We have to apply the distance matrix to that. So this distance matrix, this distance function, what this is going to give us is the distance between all data points in this T-map, this scaled matrix that we have. This takes a little while to compute. That's pretty quick. So if we look at the dimensions of this, gives me null. Let's see what we have here. So this gives us, we're doing this by samples. I forgot about that. So there's only 8 samples here. So this is going to be the distance between the subjects in the data set, in the columns. So this is going to tell us how similar is subject 1 to subject 2 to subject 3 to subject 4. And then we do the hierarchical clustering. I'm saying method equals complete. This will do bottom-up clustering using that complete method. Getting the labels by group, getting some colors here. No, I'm not using these colors, and then plotting this, okay? And this is where I made a mistake. So I was looking at this, and I noticed that, okay, clearly, first we have 2 clusters where we have 4 on one side and 4 on another. You would think that these would be the treated and these would be the untreated, or vice versa. That's not what this shows. This shows we have treated, untreated, untreated, treated, treated, untreated. So there's a mix of treated and untreated in here. And I was like, God, that's really funny. Especially because down here, when I said, all right, let's do 2 clusters, how does that separate by treatment? It actually separates perfectly by treatment. So something was wrong when I was looking at this. So had to go back. This is what I figured this out right before we started today. I always figure these things out right before you start, doesn't it? Like you get when you're going over something, you're like, oh my gosh, this is something's wrong here. So being a little too fancy here, I wanted to add these labels so we could see what data points correspond to what people, and that's where things went wrong. And you can see that I'm using this order here. So I'm taking the hierarchical clustering and I'm using $ ordering. And if we look at how this hclust procedure works and what it gives back, it tells you that the ordering is a vector giving the permutation of the original observations for plotting, in the sense that a cluster plot using this ordering and matrix will merge, will not have crossings of the branches. So what this means is basically this ordering vector is giving me the order of these points at the end. Okay, so what does the ordering vector tell us? Well, it tells us that the fifth sample is the first one. The first sample is the second one. The third sample is the third, and the seventh sample is the seventh. So let's look at these. The fifth sample, 1, 2, 3, 4, 5, is this one right here. The fifth sample is untreated. So it's a control. Here, we can clearly see that this says treated. The first sample, also untreated. The third sample, also untreated. And the seventh sample, also untreated. So these first four should all be untreated. The second four should all be treated. And then I went a little further down here. So it was this label part where the mistake was being made. So the code I was using was correct. It was giving me the labels in the order that I wanted them. But then apparently this label function, maybe it does this on its own. It uses the order. So this part was not necessary, is basically the short and skinny of it. So when I recreate the plot after removing those brackets, we can see that those without treatment go to the left, those with treatment go to the right, and we can all breathe a sigh of relief that everything worked out here. So some interesting things looking at this beyond the separation of treatment is the cell types. So these labels have the treatment, they also have the cell types on them. And this first cell type, the N080611, N080611. So this cell type is the first one to separate itself from both the treatment group and the untreatment group. Here, the second one to separate itself is also the same cell type for treated and untreated. And the two most similar are treatment by these particular cell types. I thought that was kind of cool that you can see both of these facets from the hierarchical clustering. And that's kind of a neat thing about it, that if we're just doing k-means, we don't get to see the evolution of the clustering like we do here with hierarchical clustering. Okay, yeah, and we can see that these go by group as expected. Here, I did end up using 200. I was going to use just 50 for this one, but I used 200. So yeah, when you get large numbers, things get pretty wild. So it's actually hard to see how this breaks into 3 groups, but we can certainly see how it breaks into 1, 2, 3, 4 groups. One group right here, one group in the middle, one group here, one group here. And we can see the size of the groups, 1, 2, 3, and 5. the so one thing that we'll take a look at in a second is using this. Sorry, it, the the heat map we created on day 12. This did hierarchical clustering on both the rows and the genes, both the rows and the columns, so the genes and the samples, and then ordered things in that along those edges on both ways, which is kind of a neat thing you can do. We can also compare these results, the k-means and the hierarchical. So comparing clusters is a challenge. One of the things that makes it challenging is the numbers don't mean anything. So the fact that something was in cluster 4 for hierarchical does not mean it's going to be in cluster 4 for k-means. It could be in any of them, even when things are going exactly right. And also, they're not going to always match. So what do we see when we compare these two? So we have the hierarchical clusters on the top, the k-means clusters on the side. So that k-means cluster 2, which is pretty large, that appeared to have some good agreement with cluster 4 in the hierarchical results, and that is going to be this middle cluster, which is cluster 4. So most of those were from cluster 2 in the k-means algorithm. What else did we have good agreement on? Cluster 4 in k-means was mostly in cluster 1 of hierarchical, but 1 of hierarchical also had a bunch from 1 from k-means. And so, hard to really differentiate there. Cluster 3 from k-means mostly went to cluster 3 of hierarchical, but again, k-means cluster 1 kind of got spread out among a bunch of things. Cluster 2 in hierarchical really didn't have much agreement here. It's a small cluster that's really seems to be pretty spread out, and maybe that's that one there. I'm not sure. We'll compare these. We'll compare these again in the heat map. So the heat map we looked at previously. I'm just going to run this in R so we can look at this a little bigger. I go down here. There we go. Okay. All right, so here, again, we're looking at the top 200 most variable genes. On the top, we have the hierarchical clustering by sample. What are the colors down here? This second color bar is the treatment, treated or untreated. The top color bar is the cell type. We can see that pattern that we had before of how those separate. On the row, we see the dendrogram. We see how these are clustered. The colors here correspond to the k-means cluster, which now I've changed because we redid the k-means analysis. I've changed it to 3, so this actually looks a little different than what we had than what's in the HTML file. The HTML version did this with 4 clusters in the k-means, which it was driving me crazy because I could not tell the difference between these two colors. So I'm actually glad we changed it to 3. So what do we see here? This top group, which is mostly cluster 1 from the k-means algorithm. Some of cluster 1 is also sprinkled in down here, but it's mostly cluster 1 from the k-means algorithm. We see a difference between the treated and the untreated for most of these. So more red over in this region, more blue over in this region. This second cluster here, which is mostly cluster 2 from the k-means algorithm. We have more blue for the untreated, more red for the treated. So we're seeing some probably differential expression here. Remember, these 200 genes are not the most differentially expressed genes. These are the most variable. If we look at the most differentially expressed, there'll be a very clear red, blue, blue, red type pattern. What we have in the bottom here is a little harder to differentiate in terms of this group and the dendrogram. Most of these are in cluster 3. We do see some others that are sprinkled in from some of the other clusters, though. And it's not quite as clear. Honestly, it looks like they are similar by cell type. So over here, similar to what we have over here, these are the same cell type. The second one, same cell type is over here. Here we are seeing something similar. So these genes may be those that show more differences by cell type. That may be popping up. Now, of course, that's nothing that would come up in our differential expression analysis because we're controlling for that fact. But so it's relatively interesting. I think the top one's probably the hardest to differentiate. I think this middle one is more clearly differentially expressed genes. This bottom seems to be genes that pick up the differences in cell type. I don't know what the top one's doing. Maybe there's a little similarity by cell type there, but not as clear to me as the other two are. Anyways, it's they're interesting things to look at. So we can also look at the silhouettes that we looked at earlier. We can also look at these to evaluate the quality of the clusters. So here, this is the k-means with when we had 4 clusters, looking at the silhouette width. So clusters that have higher silhouette widths are more close to one another. This gives the average silhouette width for all the clusters. This also leads us to say that cluster 2 seemed to be the best one that we had. So that's information that we can use. We don't have to pay attention to all these clusters. We could look at the ones with the highest average silhouette width that are ones that have the highest quality, and we could redo this for this situation where we have 3 genes. Here, yes, cluster 2 is still the highest quality, and cluster 1 and 3, they don't hold together quite as well. So what does this show? Well, each are genes that fit very well within their particular clusters, short or negative bars. The gene is ambiguous, and we can't really tell where it goes. If it's negative, it's almost closer to another cluster than it is to its own. Average widths around that 0.1 to 0.2 mark are going to be pretty common when we're doing genomic data. This doesn't mean the clustering's wrong. It just means the clusters are not perfectly separated in this full high-dimensional data that we have. Okay, so we're running up on lunch here. Little different, some comparisons between the k-means and hierarchical methods that we went over. The hierarchical method is going to be a little bit more computationally intensive, but not overly so. Other thing I wanted to go over, clustering it's exploratory. It's not inferential. We don't get p values from clusters. It's a way to explore the data. It's part of exploratory data analysis, and it's really there to to guide you in your understanding of the data. As all these methods are, this is just one that doesn't have that inferential side of it. We can do clustering in separate ways, and we can look, does the same structure appear with different methods? And what we saw here, one of the main results was that, this second cluster, it holds up well in k-means versus hierarchical. Also had a high average silhouette score. It had this when we used 4 clusters. We saw the same thing when we had 3 clusters. So that's a good sign that that cluster, there's something real there. We could take a look at it. We could follow up with it. The heat map is a really nice visualization technique. Lets you look at the groups and kind of get some information about what's going on. Okay, we're finishing up here. Just some quick review points here in terms of multiple testing. What is the point of it? Family-wise error rate versus FDR, and some of those main points with clustering. Tomorrow, we'll talk about principal components analysis, something that we've used a lot when we've been visualizing the data. We are not going to get to penalized regression, but we will talk about doing regression with principal components analysis. So I'm excited to do that. In the afternoon, you're going to apply some of these techniques, multiple testing and clustering, on some of the data that we've been using in the lab there. Okay, we will break for lunch. Have a good lunch. I will see you back here at 1 o'clock. And if you have any questions, I'll stick around for a few minutes to answer those. All right, I'm going to share my screen here so we can talk a little bit about the lab for today. Here we go. Okay, we have a different data set that we are going to be considering here. This is the fission data set that we're going to use. Haven't used this one before. This is an RNA sequencing experiment in fission yeast, comparing a wild type strain to an ATF21 deletion mutant under oxidative stress. Sounds interesting. So the experiment has 2 factors, has strain, and it has time. So 0, 15, 30 minutes after the stress was applied. We are only going to use time 0 here. There are 3 wild type, 3 mutant replicants at time 0. And we're going to ask which genes differ between the 2 strains at baseline. So here's some packages that you may need to probably need to install. Here we're looking at the this is what the fission data looks like. It's already in this summarized experiment formula, which is great. This is the call data. So we have time 0. We have 3 replicates for the wild type strain. There's also 3 replicates for the other strain. They're just not shown here. So here we're going to subset this to only consider the t equals 0 time. So we don't have to worry about controlling, adjusting for time or anything like that, independent measurements and all that kind of stuff. So the nice thing about this is we are going to subset the whole study just by using this fission brackets such that fission minutes equals 0. So then the call data will only correspond to those that have time equals 0. The count data will also only have those that are at time equals 0 as well. So that's a nice thing about the summarized experiment format. Okay, so we are running this. We're giving it the fission data set. and design matrix, we're only including strain in there, setting the reference level, doing some filtering, 6,336 genes, not as many as we have before, running the experiment here using DSeq, getting the results. Little summary of the results. Clearly, there's not as many significant genes for this experiment as there were with the previous two that we considered. You can certainly see that with the raw p-values histogram, much closer to that uniformity we expect under the null. There's not that peak towards the bottom here. Okay, so the exercises. I put this in the chat. The exercises I would like you to do are exercise 1 to do A, B, and C. This will be applying the two different corrections and looking at the differences in the results of these three. Exercise 2, this is just looking at the results from this experiment, more similar to what we did yesterday. And then exercise 4, where here you're going to use this mat object. So look at exercise 3 for how this mat object is created. You're going to do some k-means clustering. Okay, how many clusters should you use? Fit k-means with that cluster, and then you're going to have a heat map similar to what we did towards the end, and look at, hey, what can we see in terms of the heat map? Do we see any patterns there? Lastly, just going to be computing the silhouette width for however many clusters you chose. Are some of those clusters, do they appear to be better than other clusters? Those type of things.